All right, welcome to this week's iteration of the LJI seminar series. And we're very fortunate today to have as our speaker Gordon Freeman. As it happens, I've known Gordon for a long, long time. But even someone meeting Gordon for the first time would find it easy to recite his biography. He started his baccalaureate studies at Harvard, got an AB and PhD there, and has been at the Dana-Farber Cancer Institute and Harvard ever since. Gordon came into immunology at a time when it was just opening up to molecular biology, and he applied his molecular expertise to quite a range of problems. But one thing that he's been closely associated with all along the way is the B7 family of proteins. As you know, B7-1 and B7-2 are co-stimulatory ligands for CD28, as well as co-inhibitory ligands in another context. And Gordon cloned B7-1 in 1989, and shortly after was the one who demonstrated that it was a co-stimulatory ligand for CD28. He followed that by cloning B7-2, and over the years he's developed a great deal of knowledge about how these two B7 family proteins function in biology and pathophysiology. These days, though, outside of the strict immunological community, the B7 proteins that are probably best known are PD-L1 and PD-L2. And here again, Gordon was in the game right from the start. He cloned those proteins, PD-L1 in 2000 and PD-L2 in 2001. And again, as with B7-1 and B7-2, he's been following up on the biology and pathophysiology over the years. And in particular, he's developed a great part of our scientific insight into how PD-L1 blockade works as checkpoint blockade in immunotherapy. Now, along the way, Gordon's research has attracted many awards and honors. Just to name a few, the CRI William B. Foley Award for Research in Tumor Immunology, the Warren Alpert Foundation Prize for checkpoint blockade cancer immunotherapy, the election in 2021 to the inaugural class of fellows of the Academy of Immuno-Oncology, and election earlier this year to the National Academy of Sciences. So his title today is tantalizingly PD-L1, PD-1 cancer immunotherapy and new pathways. So Gordon, we're very pleased to have you here, and please take over the lectern and tell us what's new in your research.
Thank you for the incredibly kind introduction, Pat. I would say that as an undergraduate, I had delusions of grandeur and thought I might be a mathematician. And I was taking an abstract algebra class, which convinced me that I was not meant to be a mathematician. And with Pat's help, I managed to get through it. But since then, I've focused on more biological science. And these are my disclosures. And what I wanted to cover today was 4 themes: a PD-L1, PD-1 overview, a new target for immunotherapy, HHLA2, the characteristics of a good therapeutic antibody, and the PD-L1 CD80 cis interaction. So we can now unleash the immune system to attack cancer. And how did we learn to do this? And what we learned is a new strategy to basically stop the tumor from turning off the immune response. And this has been termed checkpoint blockade. Now, what I've studied for over about 40 years ago now is the process and the molecules that regulate T-cell clonal expansion. At the beginning of any disease, you only have a few hundred T cells that can fight that disease. But once you get an influenza infection, you know that this clonal expansion is critical because you can feel the lymph node under your throat going to the size of a golf ball. And this is the T cells progressing from a couple of hundred to within a week a couple of million T cells, which is enough to be an effective fighting force. And there are two signals required for this optimal T cell clonal expansion. The T cell receptor signal gives it specificity, but you need a second co-stimulatory signal provided by the interaction of either B7-1 or 2, the CD80 and 86 molecules, with CD28. And this gives you the T cell proliferation, cytokine production, survival signals, and T cell help for B cells that are critical to a successful immune response. Now, after initially identifying the B7 molecules, as I started my own lab, I started asking what other molecules looked like the B7 molecules. And we identified a molecule called PD-L1. And this was a collaboration between 3 labs, my lab at the Dana-Farber, Clive Woods at Genetics Institute, and Tasuko Hanjo's in Kyoto. And in this JEM paper, with Clive and Tasuko as co-senior authors, we showed that PD-L1, we molecularly cloned it, and showed it was a ligand for PD-1, which had originally been isolated in the Hanjo lab, and that this engaged a signaling cascade. So this identified the drug target to block the interaction of PD-L1 with PD-1. And the PD-1 signals regulate the outcome of T cell receptor CD28 signal. When PD-L1 or 2 engages PD-L1, the cytoplasmic domain of PD-1 becomes phosphorylated and recruits a phosphatase, SHP2, which then can dephosphorylate the proximal signaling molecules in the CD28 and TCR signaling cascade. And this basically results in reduced T cell proliferation, cytokine production, and cytolysis. And the idea of T cell exhaustion comes from this and is a really useful paradigm for understanding it. Now, why have negative signals like PD-1? Because it's critical for maintaining peripheral immune tolerance, for tuning down the immune response after the elimination of disease. So after you eliminate antigen, PD-1 comes down. And preventing for too strong an immune response from damaging tissues. And indeed, if you have a PD-1 knockout and infect it with LCMV, it actually is a lethal disease because the immune response is too robust. Now, we originally had isolated the PD-L1 molecule from an ovarian tumor. And we really wondered whether this was a tumor molecule or whether it was an immunological molecule. And we began to look for immunologic activity and showed it turned off activated T cells. But we also looked with David Dorfman for immunohistochemistry of solid tumors, since the molecule originally came from the ovarian solid tumor. And surprisingly, we found PD-L1 expression in a lot of these tumors, which we shared with Dr. Hanjo in early 2000, after which he began his PD-L1 tumor efforts. And this was the first immunohistochemistry was published beautifully by Lieping Chen, and this was published somewhat later in 2003. Now, in our original paper, because we had this information that came from a tumor, we said that PD-L1 is also expressed in some cancer, as 3 ESTs are from human ovarian tumors, and this raises the possibility that tumors can use PD-L1 to inhibit the anti-tumor immune response, which has proven very true. And we made antibodies against PD-L1 and PD-1 and showed that in 2001 that they enhanced immune responses. So here's the, this is CD4 cells with anti-328 and depleted accessory cells. You have basically a background level of T cell activation here. If you block with PD-L1, you get an increased amount of interferon or an increased amount of proliferation. So blocking this pathway gave you enhanced immune activation. And this, we sent the transfectants and cDNAs to Dr. Hanjo, who made a PD-L1 antibody and first used it in a PD-L1 immunotherapy experiment by Yoshikawa Y in the P815 tumor model. And there, the PD-L1 antibody gave some long-term survivors. And he kindly thanked us in the acknowledgement section. So essentially, PD-1, PD-L1 blockade can unleash an anti-tumor T-cell response with increased gamma interferon production, increased killing of tumor cells. And beginning at a little biotech, Medarex, with Alan Corman, this has been translated into multiple approved PD-1 and PD-L1 antibody drugs, including nivolumab, pembrolizumab, atezolizumab, and multiple others approved in the US or China. And these have had great success in multiple clinical trials, and the PD-1 or PD-L1 drugs are now approved in 22 different cancer types. This actually corresponds to about the tumors which cause more than half the tumor cancer-related deaths in the United States. And the response rate can vary considerably, from a high up in about 80% in Hodgkin's lymphoma to a low of about 15% in cervical for the approved drugs. So there's a range of response rates, averaging 20 to 30% in most of the solid tumors. It clearly doesn't work in all patients. About 60 to 70% it doesn't work. So there's tremendous opportunity for better therapies. It doesn't work in all tumor types. For instance, there's no success in glioblastoma, pancreatic, or non-mismatch repair deficient colorectal. So there's tremendous need for better immunotherapies. One of the other reasons for the enthusiasm for immunotherapy is the durability of the response. This was first seen in CTLA-4, and this is survival of melanoma. Historically, melanoma was a lethal disease, with few survivors past 2 years. With CTLA-4 antibody, pioneered by Jim Allison, you saw a flattening of the survival curve after about 2 years. And people who were alive after 2 years were very likely to be still alive 8, 9, 10 years later. The PD-1 drugs have been around about 5 years less, but what you see is that the survival curve does flatten out with the PD-1 treatment at a higher level, about 35, 40%, as opposed to the 20% of CTLA-4. But the distinguishing characteristics is durable responses. The other distinguishing is that the CTLA-4 only works well enough as a single agent to be approved in melanoma, whereas the PD-1 as a single agent is now approved in 22 different types of tumors. The reason for the caution for immunotherapy is the diverse range of immune-related adverse events that occur, which can occur in almost any organ system. The adverse events are worse with CTLA-4, not quite so bad with the PD-1 or PD-L1, but still grade 3 or 4 events occur in about 14% of patients. Now, the future is clearly combination therapies, PD-1 blockade plus combination with other immune inhibitors like CTLA-4 or LAG-3, or PD-1 blockade plus kinase inhibitors or some chemotherapies. And indeed, the ones in red have now been trialed in phase 3 and are approved drugs, such as the CTLA-4 and LAG-3, the cisplatins, or the angiogenesis blockers. Other possibilities include CSF1, TGF-beta, radiation, HDAC inhibitors, or PD-1 plus immunostimulators, such as OX40 or CD40, IL-2, TLR ligands, STING or bispecifics, PD-1 plus cancer vaccines, oncolytic viruses. CAR T cells become exhausted, and we showed that putting a PD-L1 antibody into a CAR T cell, we showed with Wayne Marasco, would armor the CAR T cell and give it a much longer effective lifetime. Or there can be PD-1 blockade plus microbiota or nutrients. So there's really a wide variety. But despite the initial enthusiasm, and there are now over 300 agents which in the literature have been shown to work with a PD-1 antibody in a mouse model, a lot of these, or all of these, will not work. And this includes IDO has failed, a phase 3 trial, or MEC, or CSF1R. So the challenge now, and there are over 7,000 combination trials now ongoing in patients, is to test which of these combinations will effectively work and be safe. So I wanted to now turn to what additional checkpoints can be targeted for cancer immunotherapy. And this is the HHLA2 team. And I wanted to highlight Rupal Bhatt, Abdallah Burgess, and Kathleen Mahoney, who've done a lot of this work. And what we've done is basically followed the B7 tree down the homology ladder. Liping Chen has focused on B7H3, Xing Zhang and Liping on B7H4, and we began to look at another molecule called HHLA2. And this was known to have an activating receptor called TMIGD2, isolated and characterized by Liping Chen and Xing Zhang. But surprisingly, the in vitro activity of an HHLA2 protein was primarily inhibitory. So the biology wasn't completely understood. We asked what could be the inhibitory receptor for this pathway. And there are multiple immunomodulatory B7s that can be expressed by tumors. These, of course, include PD-L1 and L2, H3 and H4. There are 30 members in the related butyrophilin family, the VISTA molecule, ICOS ligand, and what we focused on was HHLA2. And basically, my son is a computer bioinformatician, and I got him to basically look at renal cell carcinoma for what was the expression of all the B7 family members in RCC, and how did it compare to normal kidney. And what we found is that PD-L1 and PD-L2 were increased a little in the tumor, but it wasn't remarkable. B7H3 went up. B7H4 actually went down. BTNL2 unchanged. VISTA went up. But the one that went up the most in the tumor was HHLA2. And so we really began to focus on that molecule. And we made antibodies that worked in immunohistochemistry. And here you can see that the immunohistochemistry shows nice cell surface expression in sub-kidney cancers. It certainly wasn't expressed in all kidney cancer. Here's one with a nice negative signal. And what we did is we looked for expression of PD-L1 and HHLA2 in kidney cancer. And basically the y-axis is how much PD-L1 is in that tumor. So this tumor has a whopping amount. These have pretty good amounts. The ones down here don't have any PD-L1 or very low. But it turns out these tumors have quite a bit of HHLA2. And lots of them have HHLA2. If there was co-expression, the dots would be up here, and you don't see them. So they're really not co-expressed. And they're really very differently regulated. Everybody knows interferon upregulates PD-L1, and this is really shown here in this tumor cell line. Gamma interferon will upregulate PD-L1, but gamma interferon doesn't at all upregulate HHLA2, nor does IL-10 or TGF-beta. So they're really very differently regulated. And actually, we did find rare tumors which co-expressed. But when you look at them for where the expression is, here's staining for HHLA2 or staining for PD-L1. What you see is that the HHLA tumor expression is down here, but the PD-L1 expression is up here. There's no HHLA2 expression in this area, no PD-L1 expression in this area. And this is just higher magnifications. So even in a tumor that co-expresses on a global level, the expression is in different areas. So we're really now actively investigating what in vivo signals upregulate expression. And so we also asked, what is the inhibitory receptor for HHLA2? And we did a screen with retrogenics, which basically has a system of 5,000 cell surface cDNAs individually transfected into 293 T cells. And in the primary screen, we saw a signal with a molecule called KIR3DL3. And KIR3 is a member of the killer inhibitory receptor family, of which there are about 15 members. So we individually spotted each family member here in a secondary screen. And what you see is where there's a black signal, there's binding of HHLA2. And it binds well to the KIR3DL3 transfected cells or to the positive receptor, the TMIGD2. It doesn't bind to any of the PD-1 or PD-L1. And this is the different members of the KIR family. Most are characterized by binding to MHC molecules, but the KIR3 was rather a mystery. It was that these are encoded in one region of the chromosome, and the KIR3 is unusual in that it's at the end of the group of genes, and it's never lost, whereas other of the family members can be lost. So it's always maintained. It has 3 Ig domains and 1 ITIM motif, so consistent with a negative signaling effect. And we've published this, as also has we and Xingxing Zhang's lab, that there's basically an inhibitory interaction between HHLA2 and KIR3. And this basically looks at expression. The KIR3 is a pretty rare molecule. It's not as common as PD-1. It comes up after a couple of rounds of stimulation in CD4 or CD8 activated cells, but it takes a while to become significantly high. It's on the T cell effector memory RA cells. It's high there, which are a good proportion of the intratumoral cells. And it's on intratumoral NK cells, the CD56 DIM cells. And we looked at the affinity of the two receptors, because in the B7 family, CTLA4 has a much higher affinity than CD28. But in this family, the affinities are about the same for either the TMIG or the KIR. And so what we think is in the lymph node, probably the TMIG is more important as a stimulatory molecule, but in the tumor, the KIR3 is dominating and giving T cell suppression. Now, we wanted to test the biological activity of the KIR3DL3 molecule, and to do that we made a Jurkat cell which expressed KIR3, and an artificial antigen presenting cell, a CHO cell, which has an anti-CD3 single chain FV on the cell surface, which is going to bind to CD3 and give a T cell activation signal. And we co-transfected HHLA2, which is going to interact with KIR3. And then we tested this model, giving CD28 stimulation with just CD28 antibody. And we have an IL-2 promoter with a luciferase readout. And what we see is that without any HHLA2 there, the system gives a strong IL-2 production signal. But that if you put in the HHLA2 in the system to give a KIR3 signal, we turn that signal off. So we made HLA2 and KIR3 antibodies that'll have different properties that will block or not block, or block only one of the ligand receptor interactions. And what we hypothesized was that antibodies that block the inhibitory signal through KIR and preserve the HHLA-TMIG stimulatory signal will be the more effective immune therapeutic agents, because the binding site for these two is on the first domain, relatively close to each other, overlapping but not identical. And what we see is that in this Jurkat cell system, again, the HHLA2 will lead to an inhibition, but that if we have a double blocker, it will give us an enhanced amount of IL-2 production, and the single KIR selective blocker will give the greatest amount of stimulation of the T cells. We also tested whether this would enhance NK cell lysis, and what we see is that the antibodies that block, this is the control level, they will enhance lysis. Here you can see a good enhancement by blocking antibodies. Antibodies that don't block the interaction don't enhance. So we think this is an interesting system to explore, and we have some real-world evidence that this may be important. This is basically overall survival after PD-1 therapy with nivolumab in RCC, and the survival is best in the PD-L1 high, HHLA2 low cells. So here's the survival. If this tumor expresses PD-L1, it's more responsive to PD-L1 therapy. But if the tumor if it expresses both PD-L1 and HHLA2, it does very poorly, the red line here. If it expresses PD-L1 with no HHLA2, those are the tumors that do the best with PD-1 therapy. And we see HHLA2 expression on a pretty wide variety of tumors, very strong on renal and pancreatic, some on prostate, on lung adenocarcinoma, and colorectal. So we think it's a possible target for these tumor types. And Zhang's lab has also shown that this is a similar situation in lung cancer. And in lung cancer, about 29% of patients will express PD-L1, and about 47% will express, 63% will express HHLA2, with only a small 16% overlap. So there's a lot of patients which have HHLA2 and not PD-L1. So we think blockade of this pathway could impact a large proportion of PD-1 refractory patients in lung cancer. So in summary of this section, the HHLA2 is a ligand for KIR3. It's an inhibitory receptor expressed on activated T and NK cells. Blockade will enhance both T and NK cell activity, and the HHLA2 is expressed in the majority of clear cell RCC, and it's non-overlapping with PD-L1 expression. And the HHLA2 is expressed in multiple tumor types, and may impact a good proportion of PD-1 refractory patients and have the possibility of monotherapy activity. You may wonder, I've looked at all these mouse RNA studies. I have never seen HHLA2 come up. And there's a good reason for that. And that is that there's no orthologs of this pathway in mice. All three molecules, the HHLA2, KIR3, and the TMIGT2, are absent in rodents. They're present in primates and some species in between. So you're not going to mouse model this. And so one of the great challenges of this system to model it in vivo is how to do it. To use humanized models or human xenograft tumor models. One would have to, the possibilities of a knock-in aren't there because there's not a homologous molecule to knock it in. And so one of our challenges right now is how to best model this in mice, which we're exploring. So another theme I wanted to go into was what makes a good PD-1 or PD-L1 therapeutic antibody. And this is 2 papers published recently from my lab and a collaboration work primarily done in Dane Whittrup's lab by Sarah Coles, and in my lab by Melissa Boo. And basically all of the FDA-approved human therapeutic antibodies against the pathway have a high affinity, they block the receptor-ligand interaction, they have low Fc effector activities, and they're same species, they're human antibodies. And so how well do the antibodies used in mouse model studies reflect these properties? And the answer is not as well as they should. This basically characterizes the binding of the 9G2 lab anti-PD-L1 antibody made in the lab. It's a very high affinity antibody, well under sub-nanomolar, and it's a very good blocker of the interaction with PD-1. So that's a good model antibody. How about the PD-1 antibodies used in studies? And we compared the 1A12 lab antibody made in the lab with the RMP114 antibody, which is also used in a lot of in vivo studies. And the first thing that surprised us was that even though both are pretty extensively published, the RMP114 has about a 2 log lower affinity. And this is blocking of receptor-ligand interaction. These are the controls that don't block. The red lines are the blockade by the 1A12, which is a very effective blocker of the interaction. And consistent with its 100-fold lower affinity, the RMP114 will block, but you need a lot more of it to have an effective blockade. Similarly, if you do an examination of biological activity using a Jerkit NFAT luciferase reporter system with, again, this CHO single-chain FV anti-CD3 system, which expressing murine PD-L1 to engage PD-1. What you see is that the 1A12 or 9G2 will enhance IL-2 activity, and they'll do it at very low concentrations. The RMP will enhance T-cell activation, but again, you need 50 to 100-fold more of it to have a good activity. The RMP130 antibody doesn't block receptor-ligand interaction, and it has no activity to enhance T cells. And if you're just phenotyping T cells, the RMP130 is a nice antibody because it doesn't block. You can phenotype cells that have a blocking PD-1 antibody there. So some conclusions from this is that the 1A12 is about a 2 log higher affinity and blocking capacity than the 114. The 114 probably is used a lot, but it may lead to a situation where you're weakly blocking the PD-1 signal, giving the combination agent a lot of room to have an enhancement. So it's probably good for publishing papers, but not good for accurately predicting what will work in human clinical trials. I don't think the RNP-114 has a different mechanism of activity, but it probably should be explored. And the RNP-130 is a non-blocker and has no effect on PD-1. Now, how good a PD-1 antibody do you need for efficacy? And this is work done by Sarah Coles in Dane's lab. And basically what she did is we sequenced the 1A12 antibody, and Sarah made a whole series of PD-1 variants with different affinities by mutation. And so she has some 1A12 variants which have very, very high affinity. This is the KD of the affinities. Some which have moderate. The parental antibody has this high affinity. And these are low affinity variants. So which ones work in a mouse tumor immunotherapy experiment? And this y-axis here is basically a model which says what's the time of PD-1 receptor occupancy by the antibody, which is above 99% occupancy in the tumor. How many days will that last for? So the standard parental antibody would saturate 99% for about 6 days. And since the standard therapy in mice is to give the antibody once every 3 days, it'll stay saturated, as will these. The low affinity antibodies won't saturate. And what you see is that all the high affinity antibodies will work about equally well. There's no statistical difference between how well a high or an ultra-high antibody affinity works. But the low affinity antibodies don't work. Okay, so you do need a good affinity at this dose. But what if you change the dosing? What if you take a low affinity antibody and give a lot of it? So we gave just a little of the high affinity antibody, but a heavy dose of the low affinity antibody. And by giving enough of that low affinity, you should bring the occupancy up above 3 days. And what you see then is that if you give enough, the low affinity antibody becomes efficacious. Now, what if you give just a little of a super high affinity antibody? Is that any better? And so here we've given the parental antibody or the super high antibody, but not very much of it. And what you see is they're both about equally effective. So there's actually a threshold of PD-1 affinity, and once you get to about sub-nanomolar, if you make it picomolar affinity, you really don't make a better therapeutic. And indeed, if you give just 10 micrograms, it doesn't work. The two antibodies have about equivalent activity. What if you give it less frequently for a super high affinity antibody? So here, it's given only once every 6 days instead of the standard every 3. And so this is how the occupancy would look, but they're both equally efficacious. So there's an affinity threshold. So the PD-1 antibodies have a straightforward receptor occupancy mechanism of blockade. You basically block to the binding site. But there's a threshold affinity beyond which higher affinity antibodies aren't more efficacious. And the maintained efficacy of the super high PD-1 antibodies demonstrates that the PD-1 antibodies don't have a binding site barrier behavior that would limit tumor penetration and efficacy. This is a phenomenon that's seen with some anti-tumor antibodies, but mostly it's antibodies that bind to the tumors. And they have the problem of they all bind right to the surface of the tumor and they don't penetrate. But because the PD-1 is on the T cells, which is a relatively rare target, you get good tumor penetration. You don't get this surface binding effect. The last area I wanted to talk about was the PD-L1 CD80 cis interaction. And actually, one more point from the last is a lot of groups are investigating bispecific PD-1 antibodies. And one point that comes from these antibody studies is that monovalent PD-1 antibodies work perfectly well. And what that would lead you to believe is that in a bispecific format, the single PD-1 antibody will give you about the same efficacy as a bivalent PD-1 antibody. So the other half of the bivalent can be occupied by an antibody to another molecule. So one of the surprises in this pathway is how many other alternative interactions there are. And I'm going to talk a little about the PD-L1 CD80 cis interaction. And multiple years ago, we showed that PD-L1 also binds to the B7-1 molecule. The highest affinity interaction is between PD-1 and PD-L2. PD-1, PD-L1 is a little less high affinity, and the B7-1, PD-L1 is somewhat lower affinity. This is similar to the interaction affinity of B7-1 with CD28. On the other hand, B7-1 doesn't at all bind to PD-L2, though PD-L2 does have another interesting alternative receptor, the RGMB molecule. Now, we initially had thought of this interaction as occurring the same way PD-L1 did, that PD-L1 or B7-1 would interact with PD-1 on the cell across from it in a transactive system. And Yanping Xiao in the lab basically tested this. And this is the reason you do experiments where you think you know the answer. And what this is we've made transfectants with PD-L1, B7-1, PD-1 or CD28, and we've labeled one cell with a green fluorescent dye and the other with a red fluorescent dye, and basically mixed them and then run them through a cell sorter and asked which act like they're conjugates. So a conjugate will have a high forward scatter. So these are the conjugates out here. On the other hand, here are the single cells. And what you see when you look at this is that the B7-1 PD-L1 cells don't interact with each other. There's no conjugates. On the other hand, PD-1 with PD-L1 form excellent conjugates, or the B7-1 CD28 form excellent conjugates. So it's not a failure of affinity. They don't bind across. So Apurvi Chaudhary in the lab tested whether they bound in cis. And the system she set up was to basically do a nanobit proximity assay. And this assay basically puts 90% of a luciferase molecule called a large bit on the PD-L1 molecule, and the B7-1 molecule has 10% of the luciferase, which is called a small bit. Now, the B7-1 molecule is a natural homodimer, as shown by the crystal structure. And basically, this is consistent with this. If you put the large bit on the B7 and the small bit on the B7, you see that the two B7 molecules will interact in a cis interaction. If you put the large bit on the PD-L1 and a small bit on the B7-1, which is what's shown here, you also see a cis interaction. So that molecules can interact in cis on the same membrane. And the controls are low. So when you come together, you get the whole molecule and light. So we explored this and asked, what does the cis interaction block? And what we found is that when PD-L1 binds to B7-1, we asked, how does it affect the binding of PD-1 to PD-L1? And basically it acts as an inhibitor. So the cis interaction will keep the PD-L1 from interacting with PD-1. We made an antibody 2H11 that'll block the PD-L1 B7-1, but not the PD-L1 PD-1, and the 9G2 antibody, which will block both. And we showed that in a diabetes model, in the development of non-obese diabetes mouse model, that both antibodies will precipitate the rapid development of diabetes. So in this case, the net effect of the interaction is immunoinhibitory. And basically, this summarizes the dynamic interplay between this system. And there's essentially the PD-1 and L1 molecules will interact in an inhibitory fashion. Now, the CD80 molecules will form a homodimer and interact with CTLA4. And you can see that because it's a homodimer, it can sort of form a lattice with multiple CTLA4s. And this strong lattice, actually, David Sanson has shown, will lead to this CTLA4 literally ripping the CD80 off the surface of an antigen-presenting cell in a process called transendocytosis, which will essentially remove the co-stimulatory signal. Now, I've tried to highlight the interaction surfaces in either red or blue on the CD80. So the blues are the homodimerization system, whereas the red is the interaction with CD28 or CTLA4. And you can see that the PD-L1 CD80 interaction removes the PD-L1 interaction from PD-1, but it'll let CTLA4 still interact, or it will let CD28 still interact. And further insights from the Okazaki or Hui lab by Suguri or Zhao have shown that basically disrupting the CTLA4 lattice structure leads to poor transendocytosis of CTLA4. So you don't remove this co-stimulatory CD80 signal, but you still can retain the CD80, CD28 signal. So basically PD-L1 is a competitor which is removing the PD-1 signal, but still, and lessening the CTLA4 signal, but still leaves a good CD28 signal. So this interaction favors the CD80, CD28 signaling, less PD-L1, PD-1 signal, and less CTLA4 transendocytosis. So in wrapping up, I wanted to say that I think the future of cancer immunotherapy decisions is really going to be personalized medicine combined with immunotherapy. There's real value in sequencing every tumor. Every tumor should be sequenced for tumor mutational burden, because if there's a mismatch repair deficit, these patients have a very high likelihood of responding to PD-1 immunotherapy. It's also worth sequencing, because this will reveal whether there's PD-L1 or PD-L2 genome amplification. This occurs in almost all Hodgkin's lymphomas, but about 1 to 2% of all tumors. And Margaret Shipp and Philip Armand at the Dana-Farber have shown that these tumors have very high response rates because they're addicted to the PD-L1, PD-1 pathway. They're really dependent on it, and if you block it, you get very high responses. There's also good responses if you have a viral antigen. That's what the tumor immune system would most like to recognize as a foreign antigen. So there's good responses, as shown by Paul Neum, in Merkel cell or HPV head and neck. Its sequencing will also identify actionable mutations and identify what targeted kinase or cell cycle inhibitors are going to work in that tumors. And they'll also, of course, identify the neoantigens that are possible for cancer vaccines, as explored, for instance, by Steve Schoenberger here. You also want to profile the tumor microenvironment and identify how is the tumor turning off the immune response. How much PD-L1 expression is there, HHLA2, what are other mechanisms of immunoinhibition like IDO, TGF-beta, B7H4, matrix expression, how much immune infiltrate is there? There's advances in vivo imaging, and we can begin now to see and visualize PD-L1 expression in the tumor, or if the CD8 T cells are infiltrating the tumor in a patient. There's also advances in analysis of tumor DNA in the blood, which can tell us a tremendous amount about what the mutations are and what the T cell repertoire that's being expanded is. And this will really help us identify what's the best immunotherapy and how to choose the best targeted therapy or vaccine combination. It's really a great time to be an oncologist or researcher because we can do better. PD-1, PD-L1 blockade really works on a wide variety of tumors. There's, at this point, a moderate percentage of responders, but it has a good safety profile, and it really gives us a foundation to build on and add to. And with this success, really human creativity has really been unleashed. And I'm really optimistic because we're really learning how to do better. You really have the opportunity to advance this field and do good work. There's really a tsunami of data coming. There's over 7,000 PD-L1, PD-1 combination trials ongoing. And we're really going to learn what works, what's safest, how it works, and who it will work for. Over the years, these are the members of my lab that have really done these studies, really beginning with Julia Brown and Kwafeng Kai, the HLA work with Tony Arrola-Donovan, Kathleen Mahoney, and Abdullah Burgess, Sarah Klein with glioblastoma models, Shabu with phosphorylation of PD-L1. And it's really been a collaboration with multiple groups over the years. It's always a collaboration with Arlene Sharp's lab at Harvard Medical School on understanding the in vivo activity of these pathways, with Vijay Kuchar on the TIM3 pathway, with Arne Friedman and Lee Nadler in the original identification of the B7 molecule, Kathy Wu with vaccines, David Reardon with glioblastoma, Kwok Wong with lung cancer models, Glenn Dranoff with tumor models, and Margaret Shipp with Hodgkin's lymphoma, Ed Greenfield making all the antibodies that we've made, David Dorfman with the original immunohistochemistry, Andy Lichtman with early work on endothelial cells, Nino Chioka with glioblastoma, Sabina Signoretti with HHLA2, and Scott Rodig with immunohistochemistry. The tremendous work with T-cell exhaustion with Rafi Ahmed and John Wary, the antibody work with Dane Wittrup, early work on signaling with Vicky Busiadis, HHLA2 with Rupal Bhatt, and really the PD-1, PD-L1 field got opened up with our work with Clive Wood and Tasuko Hanjo. And I just wanted to close by saying my mother died in about 6 months from lung cancer. She was a heavy smoker. And it's just a tremendous thrill to be introduced to patients like Barry Nelson at the Dana-Farber, who's a non-small cell lung cancer patient. And he's still doing fine 9 years after beginning PD-1 immunotherapy. So the real winners here are patients and their families. Thank you.
Could you repeat any questions for the Zoom audience?
Sure.
I found the cis interactions very interesting. Is there anything controlling that? Any kind of mechanisms that, or is it just affinity and protein expression?
It's going to.
Please repeat the question.
Oh. Is what factors are controlling the cis interaction? And the answer is expression levels are what is going to regulate it the most. One side effect it has is that you can have a cell which expresses PD-L1, and if your B7-1 level is high, it'll actually hide all the PD-L1 from phenotyping with a PD-L1 antibody. This actually can mislead some of the immunohistochemistry, because some of the immunohistochemistry antibodies look at extracellular epitopes, which may be blocked by this cis interaction, which gets fixed by paraformaldehyde, whereas other of the immunohistochemistry antibodies look at the cytoplasmic domain. And we, and I think Genentech, has shown that this is probably a preferable immunohistochemistry antibody because it's not affected by the cell surface interactions. I don't know of other factors that would regulate it, though I might expect the, there's a tetraspanner, the CMT6 molecule, which sort of protects PD-L1 from ubiquitination and degradation. I would expect that will regulate PD-L1 interactions and stability.
I guess an imitation of speculation. Gordon, why do you think PD-1, PD-L1 blockade doesn't work in some cases, given the universality of upregulation of PD-1 when gamma signaling occurs?
Probably two major reasons. One is failure of the immune cells to get into the tumor, the immune desert idea. There's certainly tumors where the T cells stay on the periphery, don't get into the tumor very effectively. The other major mechanism is alternative immunoinhibitory pathways. Really expansion of myeloid inhibitory cells, many more MDSCs and M2-like macrophages dominating the T cell response and turning it off by other ways. The miracle is that blockade of just one of these pathways can be as effective as it is.
Gordon, have you looked at KIR3DL3 expression in cancers relative to prognosis? Have you looked by immunohistochemistry, or is there RNA-seq data to indicate that tumors might be enriched for cells expressing that, particularly when there's a poor prognosis against it?
We haven't explored that thoroughly yet. The immunohistochemistry says what the flow cytometry does, that KIR3DL3 cells are pretty rare. So I think to have an effect, this has to be a strong regulatory cell.
Has anyone looked in the kidney, in the clear cell kidney cancers even?
We've looked and found some, but not a lot of positive cells.
Can there be other inhibitory receptors or ACHL2?
The question was, could there be other inhibitory receptors for HHLA2. I've learned not to say there are no other ligands. I think the VISTA molecule is one where there's more and more receptors as you go on. We did, a very broad screening for HHLA2 counter receptors, and these were the only two that were identified. So I don't think there's a high likelihood, but I certainly wouldn't exclude it.
What about the expression of the cure3 molecule on T regulatory cells, Treg cells?
Is the KIR3DL3 expressed on T regulatory cells? Not significantly, no. I think just thinking about regulatory cells, one of the interesting things about the PD-L1, PD-1 pathway is the activity of PD-1 on T regulatory cells. And this is like work done in conditional knockout mice by Arlene's lab. And what that shows is if you conditionally knock PD-1 out in just a Treg, what would you expect would happen to the activity of the T regulatory cell?
Well, the treatment with the PD-1 antibody increases the activity of Tregs, isn't it?
Yeah, you actually, if you delete or block PD-1 in a Treg, you actually get a more suppressive T regulatory cell, which actually makes us realize that when you give a PD-1 antibody, you've got two competitive immunological activities going on. You're getting more efficacious conventional T cells, but you're also getting more active Tregs. And sort of your outcome is a balance of the two. And there's actually a phenomenon called PD-1 hyperprogression. And there's a group that's looked at that. And actually, if you have a lot of PD-1 positive Tregs at the beginning of treatment, the PD-1 blockade actually lets the Tregs get ahead. And you can actually have a bad outcome. Oh, no.
Other questions? Well, thanks, Gordon, for a terrific seminar. We're really glad you